We have five distinguished panelists, each of which is going to present for 3 to 5 minutes, and then we'll open the floor up for general questions. Our first presenter will be Dr. Daniel Adams, who's Chief Executive Officer of Protein Sciences. Dr. Adams, do you want to speak? Why don't you go ahead and speak from the chair, or would you prefer to speak from the podium? We can do it either way.
I can, either way. Is this mic working?
Yeah.
Okay. Good. So I can't go to school and the other guys, so I'll say a few things about our system. And basically we make flu vaccines in insect cells. We own the IP around that. And we start with the sequence that we get off the CDC website. We make that into a clone the baculovirus to code for that sequence, and we infect our insect cells with that. And it's fairly simple, although it's taken us only 12 years to kind of get this thing perfected. But we harvest the bioreactor, we centrifuge, we do some filtration and column work, and we come out with a highly purified protein, and that's what we do. We can, we normally will make up a master virus bank well in advance of any kind of outbreak. We have hundreds of these frozen away, and that saves us a lot of time. So, you know, we can actually be in production within a couple of weeks of pandemic outbreak from the time that we're told to get going, because our master virus banks are in general ready to go. A couple of general things. I think we heard this morning about cell culture vaccines, and our friends from the pharma companies seem to like to lump these together, but they're really, there's no relationship between an MDCK cell or a Vero cell process or a Percy-6 cell where you infect with a live virus versus cell culture that you use the genetic code. I mean, it's true that, you know, when you move to mammalian cells, you can certainly improve things over eggs, but that's, you know, that's only an evolutionary thing. You're still dealing with trying to get a virus to grow in a substrate. You're still dealing with having to adapt the virus to grow. And I know that from experience talking to people that you do have to adapt these viruses, and that still leaves you with a vaccine that may or may not be accurate to what's growing in the field. When you use the code, which you get from CDC, you're always going to be accurate. And the second thing is that, and you know, egg is in the installed capacity. So the government's got to go with eggs because that's what they got. But the fact is that eggs is not a solution for the rest of the world, and stainless steel, recombinant products, cell culture is the way that you can get things into other countries. And you know, whereas we may be able to talk the Dutch and the French into allowing, you know, into giving us some H1N1 vaccine, Robin Robinson was less than definitive in saying that they were, yeah, sure, we'll sell it to you. And it's just like Obama saying, well, you know, after we had 600 million doses and we didn't need them, he said, oh, well, we'll just give these things away to the rest of the world, but not until we didn't need them. So I think, you know, we've got to have modern technology in manufacturing. Eggs is not the way to go for, and you know, if we don't protect the Chinese, what are we going to buy? We don't make anything here anymore. So I think the key for these vaccines is that they're accurate antigenically. It's very quick for at least some of us to get them rolling out. They're highly purified, so you don't have to worry about thimerosal and antibiotics and junk. And you know, you can manufacture them in a relatively inexpensive facility. Our facility, to turn out a 60 million, you know, seasonal vaccines per year, would be in the neighborhood of $40 million. Which is, you know, a fraction of the $1 billion that Novartis spent down in North Carolina for the MDCK cell vaccine. So there's just a whole difference. Now, you know, I believe we're going to get approved this year. We are to the final stages. We've had our final meeting with FDA. You know, we're well into the red zone, and anybody who's tried to get a product approved, you know that I call it the red zone at FDA. You know, the defenses stiffen up. And, you know, it's all, you know, 3 yards in a cloud of dust, Woody Hayes football, and, you know, short passes. But, you know, no Hail Marys, no long bombs anymore. It's really it's a tough go at the end. And so it's very much like football. But we do expect to get approved this year. We do expect that it'll be easy to approve a pandemic strain as a seasonal change for us, and you heard some comments from Wellington Sun this morning that maybe if it hadn't been an H1N1 strain that was the pandemic candidate, like an H2, you couldn't have used the seasonal strain, but we believe that because ours is just the genetic change in the baculovirus, that it'll be much easier for us to get pandemic strains approved. So from every standpoint, and again, I don't want to run down eggs because they've been on the market for 60 years. They're safe, they're pretty effective, but when you look at the world as a whole, and even the US, it's not the long-term solution.
Great. Thanks, Dr. Adams. Next panelist to speak will be Dr. Rahul Singhvi, who is the Chief Executive Officer for OverBacks. Dr. Singhvi? Yeah.
Thank you. Can you all hear me okay?
Yes.
Great. So, so I represent OverBacks, and our technology has the same manufacturing system of insect cells, and we use a recombinant version of a recombinant technology called virus-like particles. So instead of just expressing one protein, as Dan's company does, which is hemagglutinin, our technology is to express 3 of the proteins of influenza: hemagglutinin, neuraminidase, and matrix, which when co-expressed self-assemble themselves into a particle that looks very much like the influenza virus, except that it doesn't contain any of the genetic materials, and so it doesn't have the capability to replicate and grow. But to the immune system, these particles appear as if they're seeing a natural infection. And so the theory is that this particle is much more immunogenic and is able to elicit an immune response very similar to what a virus would. So we've applied this technology to influenza, and I think one of the key questions was like how could we use this technology last year in the middle of a pandemic, and what were the success factors? So all of the points that Dan made about the speed and how we can make the vaccine faster using this recombinant technology, because you're not dealing with seeds of viruses and growing them in eggs, but starting with genetic material, is applicable to our technology as well. And what we did last year was to demonstrate that our virus-like particles could be used to produce a vaccine within a very short period of time. In this case, the cGMP batch was produced within 11 weeks. And we demonstrated not only that this technology was able to produce a vaccine fast, but we took it to the next level, and which was to test that vaccine in humans at a very large scale. And the key success factor for us to be able to, there were a couple of important things. One was that we had significant clinical experience prior to last year with our virus-like particles in influenza strains, both with H5N1, a pandemic strain, as well as with seasonal strains. So that allowed us to take advantage of all of that data and submit it to a regulatory authority, in this particular case in Mexico, which they saw as relevant, and they gave us permission to test our vaccine in Mexico in humans in a dose ranging plus an expanded safety study involving 4,500 people. For us to get that opportunity to test the vaccine in the middle of a pandemic was a very significant opportunity, which we took very seriously. We produced the vaccine on our own and did the clinical trial in Mexico. And thanks to our partners in Mexico who worked tirelessly and gave an incredible amount of effort, both in terms of time and money, to help us see this through, a trial that we started on the 19th of October and completed the first phase with 1,000 patients within 2 months. So we had results actually back in December, where we showed that this new technology in the middle of a pandemic could help people, because we got data that showed that the product was well tolerated and it was immunogenic. We have since then completed the expanded safety and completed the full 4,500 subject study, And I'm very, very happy to report that we have not seen a single vaccine-related or even remotely related serious adverse event thus far. And our immunogenicity data still holds very well. And so we've really taken this to the next level, where we've not only shown that the technology has promise in creating a vaccine faster, but we've created a product that has shown to be viable in a very significant clinical study. And we've submitted the data to the Mexican authorities for registration. We hope that we will hear from them very shortly, and like to get the product approved in Mexico, and hopefully supply the product if there's a need. So that's the story that I wanted to tell you, is that here's is a technology that has moved on beyond the very initial proof of concept to hopefully becoming much more of a reality.
Dr. Singhvi, thank you. The next panelist to speak will be Dr. Robert Becker, who's Vice President of Business Development for Vaccinate. Ron? Yep.
Could
Do you want to speak from
Yeah,
the...
it's okay. If you can just,
I can advance them.
you can just advance it. Okay, great. So first slide, please. The next one?
Maybe
Perfect.
There we go.
There we go, okay. So Vaccinate is a vaccine company, biotechnology vaccine company. Technology originally came out of Yale from Ruslan Mezhnatov and Richard Flavell. And the core aspect of the technology is to engineer vaccines as recombinant proteins in biopharmaceutical grade proteins that stimulate simultaneously both the innate immune system and the adaptive immune system. And one of the reasons that's important is because really the strong stimulation of an adaptive immune response resulting in neutralizing antibody in part depends upon the innate immune system recognizing the vaccine as a foreign microbe-derived entity, and then helping the adaptive immune system, in fact, respond in a very robust fashion. The way we've done that is by linking two components, one being the protective antigen that the adaptive immune responds to. In the example of flu, that's the HA molecule, the hemagglutinin antigen, which is the identified most protective of the antigens and the basis of licensure of almost every vaccine for flu that's out there. And the other portion is a molecule known as flagellin, which is recognized by a group of receptors known as the toll-like receptors, and specifically toll-5, as a component of a microbe. And when it recognizes that, it sees the vaccine itself, even though it's a pure protein in solution, as a component of a microbe, and hence triggers very strong immune responses at low concentrations. So one of the interesting aspects of this as well is we've been able to manufacture for the first time both the HA component of the vaccine and the flagellin all as one molecule, but not in eukaryotic cells, but rather in E. coli, which dramatically improves the speed at which we can get cultures moving and improves manufacturing efficiency in a culture-by-culture basis by about 2 or 3 orders of magnitude as compared to other technologies like cell culture or especially like egg. Next slide, please. So if you look at this construct, the circled components simply represent the HA molecule. We have several different versions of these vaccines today. The first one, named the one titled here C-terminus, where the HA molecule is attached. The C-terminus of the molecule has in fact gone through extensive clinical testing in young, healthy adults. And those reports, in fact, have been published, and I'll show you just a quick excerpt of that in a moment. But also, very recently, we've begun elderly studies, and in fact, on April 27th at the NFID meeting in Bethesda, we'll actually be presenting some of the elderly results that we've gotten with this vaccine, and we think those results are very encouraging and very interesting, and hopefully the public will see that as well. We also have 2 other constructs locating the globular head at different locations. One's sort of in the middle of what's known as the hypervariable region, removing what's known as domain 3 and replacing it with the HA. And the other form has 2 globular heads per flagellin molecule. All 3 versions of this now have been, we're almost done with cGMP manufacturing. of a swine origin H1N1, and all 3 versions of that molecule or of that vaccine are going into clinical trials this summer. We also have an Indian partner that likewise is evaluating the development of this product for India pandemic needs, and likewise we anticipate clinical trials in India through our Indian partner in this year. We've been able to easily transfer the manufacturing of this product to multiple parties, including our partner in India. So the product being actually tested in India was manufactured in India. The products being tested here in the US were manufactured through a CMO here in the US. Next slide, please. So when we use these vaccines, what we typically see is a potency increase versus standard flu vaccines. As you probably know, egg-based flu vaccines use about 15 micrograms per strain in their formulation. What we find is when we're at 1-2 micrograms, we're getting responses as good or better than what you see in a typical flu vaccine. This shows you some of the results in a broad dosage range, and then also when we expanded that study and focused on fewer number of doses, what the comparable results were to the placebo, to 0.5 microgram, 1 microgram, or 2 micrograms. And again, we're getting very robust neutralizing responses with these low doses. And then the elderly data will be showing you comparable results with the same kind of dosage ranges. But that needs to be discussed in a few weeks. Next slide. So, you know, I guess the point we're going to try to make here, and it's very much like the previous 2 speakers addressed, you know, in the world of pandemics, I think the one thing we learned is speed matters. And I'll simply make the point, it's not just speed to the first dose, it's speed to the last dose. So it's not just how fast can I start manufacturing and start getting doses out there. The point is, when do I get the last dose out there, right? Because if you're the last guy standing to get the vaccine, you know, if this pandemic has come through and gone before you get your product, it's as a pandemic conference from a number of years ago. They said the reality is if we don't change our technology, we'll be vaccinating the survivors. And to some extent, I think the experiences of the last year sort of reiterate that. Though fortunately, it was not as serious a pandemic as might have been the case. So the question at hand, I think, is not just when will a given manufacturer or a given technology be able to get you the first doses to go in the public. The question is when will a company or a technology be able to get you the first to the last dose, you know, in as fast a period of time as possible, so that we can in fact vaccinate and cover our populations. And you know, that's a big issue here in the US, but you talk about places like India, you talk about places in China, with not populations of 300 million people, but populations of over 1 billion people. These kinds of issues really escalate, right? And so that's why these kinds of technologies are critical, not just from a US perspective, but from a global perspective. And this diagram tries to show, based on the analyses we've done, how different technologies stack up. And I, you know, from our perspective, we can, from a sequence to actually begin manufacturing, is about a 3 to 4 week process. Again, we simply get the sequence, we make the construct, we take it into E. coli, and typically in 3 to 4 weeks, we're making purified protein. That can move very quickly into manufacturing. And because these products are highly defined biopharmaceutical grade proteins, the ease of testing and releasing these products become far greater. Meaning, again, you can get the product out into the public much faster. And again, because we're manufacturing, you know, somewhere in the order of hundreds of thousands of doses of vaccine per liter fermentation, E. coli fermentation. So actually, in a period of only a couple of months, one can readily, even with modest manufacturing facilities, not even necessarily requiring the building of a new facility, but simply using a CMO, be able to make enough vaccine to cover the US population. So from our perspective for pandemics, that's sort of the value proposition our technology and technologies like us provide. And we're, you know, actively pursuing not just the development of pandemic vaccines, but also the development of seasonal vaccines. And obviously, it's for reasons like that that we've been testing these products in the elderly, just not to talk about cost of goods and how easily you can make a dose, but also is it going to be differentiated? Is it going to be a preferred product, a product with better efficacy in these higher at-risk populations?
Thank you, Dr. Becker. The next panelist to speak will be Dr. Bill Enright, who's the Chief Executive Officer of Vaccine. And you have slides as well?
I don't.
Okay. Dr. Enright?
Thank you. Am I on here? Can you hear me okay? Okay. So appreciate the opportunity to speak at this panel discussion, and you've heard from a number of folks that have gone through some of the advantages, the basic advantages of recombinant technology. Vaxxin is taking a slightly different approach, and what we have done is utilize a very effective virus, adenovirus, to use as the backbone of our platform technology. Adenovirus is a virus that's normally found in nature. It doesn't have a lot of safety issues, normally causes symptoms like the common cold. What we've done is to modify that virus so that it is replication incompetent. So it can't replicate once it gets into the body. But it is a very effective still at getting into the normal route of the normal epithelial cells that it invades. So it's one of the best viruses. It's evolved over millions of years with humans in doing that. So we use an intranasal route of administration to deliver the vaccine, which is the normal course that the body sees this disease in influenza. And we think that offers additional advantages over other recombinant technologies in that you get a very rapid mucosal immune response followed by the systemic immune response in both antibody and cellular responses. So again, mimicking the way that the disease normally, or the body normally sees the disease, and that the body normally deals with clearing that disease. So again, a very similar process to some of the other speakers. We're not dependent on growing up the whole influenza virus. We take the sequence information as soon as it's available from the CDC and synthetically synthesize that and clone that into the adenovirus backbone, and then use the cellular manufacturing processes to grow that up into large quantities at very inexpensive because you don't have the facility requirements that you do for the egg-based processes and some of the other cell manufacturing-based processes. The cost of goods is significantly lower. And in addition, the time to manufacture this product is at least half of what traditional egg-based manufacturing processes are. So one of the other advantages to using the adenovirus as a platform technology is, as is with the case with some of the VLP technologies, or when you're adding flagellin to the molecule, adenovirus is in itself a TLR stimulant, and so it acts as its own adjuvant, so you don't have to worry about additional adjuvants. So you're only using things that are found in nature, using a piece of the influenza virus and a virus that many people are infected with on an annual basis in the United States and around the world. So we believe the combination of the intranasal route of administration, the cell culture-based manufacturing, and the self-adjuvanting properties yield some specific advantages over the other technologies in development. And we've now demonstrated that through a couple of proof of concept studies with a seasonal H1N1 study in people, as well as an H5N1 study that we completed last year, where we showed that the vaccine was very safe and well tolerated and had very promising efficacy profiles. And so we're in the process of trying to advance the technology into phase 2 clinical trials and hope to have that started towards the end of this year or early next year. And both monovalent seasonal as well as a trivalent seasonal. And we have a partnership in Korea, and our Korean partner is also working to move the H5N1 into phase 2 in Korea. Because as Bob stated, this isn't just a US issue, it's a global issue, and I think the H1N1 pandemic really highlighted this issue for a lot of countries around the world, that without manufacturing on soil, you were rather low on the totem pole as far as getting vaccine, and it may not be in time. So I think there's a renewed effort by a number of countries around the world in bringing cell culture-based manufacturing into their home countries.
Dr. Enright, thank you. Finally, Dr. Thomas Muster, who's the Chief Executive Officer and Chief Scientific Officer of Aver Green Hills Biotechnology. And
Thank you, John.
your slides are up there behind you.
Yeah, thanks a lot, John. Thanks a lot for the organizers to have the opportunity to participate in this panel here. So Aver Green Hills is in phase 2 trials with Delta flu, which is a replication-deficient intranasal influenza virus vaccine. It's a live-attenuated vaccine, which is based on the deletion of the NS1 gene, and similar, like Bill with the adenovirus-based approach, we believe that the intranasal application has the advantage to induce the protection directly at the site of the entry of the viral pathogens. Actually, we heard today a couple of talks already about H1N1, the problems associated with this. I think we were very, very lucky that it was a mild and also a very slow pandemic. And I tried to address here some points which might be used for, in which our vaccine might be used for in a kind of more severe setting. The advantages of our approach is we use reverse genetics, so that's similar to what we had from Dan and Raoul. And so we use cDNA, we construct the virus from cDNA, and actually we have a quite efficient method to generate the strains quite rapidly. Secondly, we produce vaccine strains in tissue culture, so we were able to obtain quite high yields in viral cells through different modification and adaptation of the master virus strain in viral cells. Another advantage of our approach is that some multi-dose container, multi-dose device with replaceable delivery tips is available, so which might overcome some limitations due to filling, packaging, and also to the availability of enough devices. So these are 10-dose devices. Yeah, and finally, we have quite excellent immunogenicity shown in the trials we have done so far. An example you can see here on the slide, on the left down corner, it's a trial against H5N1, and you can see the response rate, the HI response rate. Left side is the low dose, right side is the high dose. Yellow is the first dose, green the second dose, and you can see already with the low dose we get quite reasonable HI responses, and remarkable with even with the first dose, with the high dosing we obtained already 75% seroconversion, which is quite unique against H5N1, and it's even more remarkable for a live attenuated vaccine, which otherwise hardly can induce any HI responses. So this was very encouraging. May I have next slide, please? Oh, can you shift further? Sorry.
There we go.
Also was very encouraging, and what we made quite in trends in the last couple of years was that we developed our own reverse genetic systems. As you can see here, it's based on cDNA, so that's opposed to the conventional reverse genetics where you have plasmids, so we can save here the cloning step into plasmids, so we transfect directly the cDNAs into cells, into viral cells, which have been previously infected with a helper virus, or as you like, as the master strain, which has all the features of the vaccine, the NS deletions, actually modifications and mutations, which allow very high growth on viral cells. And in order to facilitate the selection process, we have a elastase cleavage site in the HA and the truncated NA. So what you do then after transfection, you harvest the supernate, and you have a mixture of helper virus and vaccine virus, and you select them by antibodies to obtain the vaccine virus. May I have the next slide, please? Yeah, a very important point is also, as we heard already today, is production, and I think that's what we all five have in common. I think we are much more efficient in production than with the conventional methods. Here you see a dose calculation in a 300-liter bioreactor. We have some assumptions here. So for example, if you have a vaccine titer in the upstream process of 8 logs, and actually this is kind of a low estimate, usually we get between 8 and 8.7 logs, or in case of the new H1N1, we got 8.4 logs. human dose, which we have 7 logs, and actually this is based on the experience we had with the previous clinical trial. So based on these assumptions, we are able to produce about 20 million doses a year with a 300-liter bioreactor, considering that we make 16 batches a year. In other words, we can produce 5 million doses in a 1,000-liter bioreactor, which is quite a reasonable amount in a quite medium-scale process. Yeah, basically to sum that up, I think we have developed a live-attenuated vaccine based on the deletion of the NS1. We have a quite rapid, efficient reverse genetics method to generate the strains, so we can produce strains in about 10 days. And finally, we have quite good yields in viral cells. Thank you much.
Dr. Muster, thank you very much. We have about 15, 20 minutes. I'd like to open the discussion up to the entire panel, and we'll take questions from the audience. Admittedly, we're up here kind of in the bleachers with the sun in our eyes, so I can't see everyone who may have a question. So I just have to rely on you back there with the microphone. If you wouldn't, Mark, to hand the microphone to whoever might have a question. I might lead off just very quickly asking Dr. Enright, your plans to get around the issue of pre-existing immunity to adenoviruses. Presumably this is a common adenovirus serotype.
It is. It's Ad5 adeno, which, and I guess over the last several years, there's been a lot of data that's been accumulating suggesting that pre-existing adeno antibodies actually don't interfere with the body's ability to mount an immune response against the protective antigens in the vaccine, in this case HA for influenza. And some data published in 2008 in PLOS ONE actually had a nice look at both intramuscular administrations versus intranasal, and in particular with the intranasal route of administration. We believe that the immune response reacts so quickly, adeno gets in and starts producing protein so quickly that it doesn't interfere at all.
Thank you.
Got a question here.
Did you take the vaccine? I have 2 questions. The first question for OverBacks. There was a review article published last year describing that the VLPs are heavily contaminated by bacteriovirus because they bind very closely to each other in an ingredient. So has that problem being circumvented, or is it not a problem at all? A second question for Aver. Okay, your AS1 mutant influenza virus is...
Why don't we take those one at a time?
Okay, all right.
It might make more sense. Dr. Singhvi, did you catch the question?
Yeah. I think you're right. I think the baculovirus and the VLPs are quite similar in size, but this is something that's a challenge that we have been able to overcome, where we have been able to separate these 2 entities through the use of chromatography and other downstream separation unit operations to a very large extent. When I say very large extent, it's 99.99%. But whatever residual baculovirus that is left is inactivated using a very benign inactivating agent. And so what we end up in the final product is a relatively pure product which has over 80% influenza protein. And the contaminating proteins are the baculovirus and the SF9 or the insect cell proteins. And that's a very conservative purity number that I've given you. Generally, we are able to get over 90% pure. It is not an easy technical problem, but it is one that we have successfully addressed.
A follow-up question is that VLPs has been produced in other systems. For instance, people now can produce VLP continuously in a tobacco plant. So have people compared the potency between different VLPs do they confer the same kind of protection? You know, doesn't matter whether it's produced in insect cells or in a tobacco.
Yeah. Well, I can't speak to other companies' products. All I can tell you is that the product that we make, we have now a clinical database of over 4,200 subjects with 6 different seasonal VLPs, so for 6 different strains of influenza, seasonal influenza, 2 different strains of pandemic influenza. And we have produced, I believe, over 25 lots under CGMPs at Novavax. So I feel very comfortable that as long as we are concerned, we have advanced this technology far enough to feel very good about the safety and immunogenicity profile of this product and the ability to make this product in a consistent manner with very different types of influenza strains.
Thank you. A question for Eva is that your AS1 mutant influenza virus is more or less similar to flu mist. And the flu mist has not been allowed to be used pre-pandemic because they worry about reassortment may actually trigger a pandemic. After the pandemic occurs, it doesn't matter anymore. So do you expect that you will have the same problem that before pandemic occurs, the AS1 mutant influenza virus vaccine will not be allowed to be used due to the potential for reassortment?
Yeah, it's a very good point. We have the advantage that we are replication efficient. So what happens here, you immunize into the nose, and the virus goes into the nasal epithelial cell but doesn't come out anymore. So the likelihood of potential reassortment is very low or dramatically reduced in comparison to a conventional live attenuated approach. But of course, it's very likely that also our approach cannot be used in a pre-pandemic situation, but once the pandemic has started.
Well, reassortment actually occurs intracellularly. So if it doesn't come out, doesn't mean it will not go out reassortment.
Okay. In this regard, we have done, although it was an animal, so some experiments. So so similar to what Bill has said, the virus induces some kind of self-adjuvant, and since the NS gene is an interferon antagonist, and the interferon antagonist is lacking in our vaccine, it induces a lot of interference. So we are not able, at least in the animal model, to once we have immunized, to subsequently infect with flu, but even not with any other virus.
Thank you.
Thanks.
Got another question over here as well.
We get the worst of it.
Hi, my name Robert Malone, Miami Research. My question has to do more with the economics that you as novel developers confront trying to produce an influenza vaccine. Let's hope that we don't soon face another pandemic. So you'll need to be able to sustain your products and get market entry into a market that I think most would agree is a commodity market with multiple very well-established producers that we've heard and surround us right now. How can you differentiate your product and gain market entry in this crowded, commodity-driven market environment for influenza vaccine?
Well, there's a piece of red meat. Who wants to go first? Yeah.
I think there's several attributes, some that are related to all recombinant products and others that are related to specific products. Recombinant products have no restriction as to the HA type that can be expressed, as you probably know, especially for egg-based manufacturing. For some strains, there's only a small portion of the human isolates that are identified that in fact will propagate in eggs. And so there's always a compromise between what's the right strain in terms of match and protection and what can be grown in eggs. That's for egg-based manufacturing. The other aspect is that when it's been known since the 80s that when one looks for generating high yield strains that will grow in eggs, in fact, there's a mutation in the HA that takes place that drops the affinity for sialic acid, so more of the virus actually remains in solution. So though nobody really wants to talk about it and evaluate it significantly, we do know that, in fact, the virus that we're producing in eggs is actually a mutated HA with a lower affinity exactly in the place where we have neutralizing antigens. So when you look at recombinant expression, any HA that you can turn into a protein, in fact, can be manufactured and very quickly, and it's going to be as wild type as, in fact, the sequence that's provided to us. So, I mean, if you ended up possibly with cell culture, but clearly with pandemic, with a better source of strains that better match what's circulating in a given year versus an egg-based manufacturer that's going to have to go with those strains that replicate in eggs, I think that just from the get-go will probably be a differentiating factor for some of these products. That's going to depend upon the CDC, though, and other regulatory bodies making recommendations for manufacturers that could make any given strain versus those strains that are provided to egg-based manufacturers. The other one is, I think, one of the key differentiators I think any of us would say is if you can differentiate your product in an elderly population, in other words, if you can get better efficacy, better responses as compared to the standard products that are out there, that's going to be a major cost driver and going to, improve both pricing and penetration in what's been considered now a commodity market. And that's why the data that people are going to be providing in the coming years regarding how these products fare in an elderly population is going to be so critical.
So Vaccinate thinks it can enter market with a more potent product that is more closely matched to the circulating strains.
I think two things. I think, and this would be Vaccinate, this, I think any of the recombinant manufacturers can make a broader range of HAs very quickly as compared to egg-based manufacturing. And as long as the regulatory authorities are willing to provide those manufacturers that can do it the best strains to be manufactured for a match to a given year, right? Those products can differentiate themselves. The other is, are any of these products either based on the dose that's being provided or on the attributes of the product itself going to have better responses in young healthy adults, but especially in the elderly population, which we all understand is the population at highest risk for infections.
And the other factor that you've certainly talked around but not directly addressed is the cost of production, which is another issue.
Yeah, I had a couple of comments. Looking at the dynamics of the flu market, commodity market, cries out for a differentiated product. So how are we going to differentiate it? Okay, Bob mentioned the elderly, and I think we're seeing, certainly we've had superior data in the elderly compared to the licensed vaccine. Second one is, 18 to 49. Don't need it, don't want it. It's dirty, it's ineffective, it gives me the flu. What a marketing challenge, right? So we don't have any of that stuff. And so there's a great opportunity to get people 18 to 49 to take a flu vaccine, and you're not going to market to these people because they're already made up their minds. 50 to 64, you get the same problem. People have made up their minds, and that's a tough place to go. But if there's anybody who can penetrate it, it's a cleaner, new vaccine. Egg allergic. A small percentage of people really are egg allergic, but if you go, I was in the food allergy business, so I know that about 5% of the U.S. population, it thinks they are egg allergic, and they don't go to the docs, so the docs don't know that they think they're egg allergic, and so there's a great market there. And just the dynamics of buying the flu vaccine, speed. Who gets hung to dry in the flu market? You're late to market. Our second largest shareholder, will be nameless, dropped out of the flu business. Why did they say? We're always late to market, we never made any money. You get killed if you're not there on the doorstep the first day. Recombinant technology puts you on the doorstep the first day. All right, Sanofi may be there on the first day too, but it's you and Sanofi you'll get the bid, and maybe GSK and Novartis, go hungry. So, there's so many ways we can differentiate our product, and I think, authenticity, but is the market going to buy authenticity? No, I don't think so. What do they know? So that's only part of it.
Dr. Enright, do you want to respond?
I think
Yeah.
specifically from Vaxxin's perspective, I think using FluMist is a good example. I mean, they were able to penetrate the market, maybe not so well initially, but I think that they've done a good job in the recent years in penetrating that market and taking a significant market share. We have what I would say is a better, safer, faster, cheaper FluMist, and so we believe that we'll be able to get the premium pricing similarly as FluMist has been able to maintain since their introduction.
Great. Right here in the front row, yes.
Jerry Dalfords with JD Technologies. Just this kind of general question to the panel, and this is more on delivery than it is to the production of the organism or the virus, because there's been a lot of talk here about nasal delivery. My curiosity here is how many of you working with powders versus mist, attenuated versus infective? Because as we all know, the nasal cavity turns over every 15 to 20 minutes. We also know the variabilities of flu mist, because depending upon the condition of the individual at the time, if he's got a runny nose, it's different than if I'm in Wyoming and I have a very dry situation going on. The other thing is how much of it is being retained in the nasal cavity versus how much is going into the lung, because when it moves past the nasal cavity, we also know that we have a completely different. And of course, that was the problem with flu mist. I won't get into flu mist. I was with Wyeth at the time. So the variability of the reaction based upon of the titer is quite high due to the nasal situation. So my curiosity is where's the industry or the panel going relative to a more uniform delivery for this? Obviously, we all want to get away from needles and injection, and if we can go to a self-administered system, obviously the market would prefer that. So
Yeah.
I'd like to hear what the panel has.
Maybe we should ask one of the live virus
Exactly. The guys
companies
doing
to
nasal.
respond, at least respond first. I'm sure you thought of it. Yeah.
Yeah, it's a very good point. So actually, we as you indicated, we compared powder with liquid. Liquid performed better. On the overall, liquid performed better, and actually, the results were more consistent. Although the results were similar, but there was a significance that the liquid is better.
What
Powder.
powder did you use?
Yeah, that's a good point. I think powder, the point we gave up powder was powder is very difficult to handle. It's very difficult to fill. It's the drying process and the characterization is quite a headache.
You have to design the powder
Yeah.
as well as.
Yeah, it might even be that we did not have the right consistency, the right particle size, but we more or less, because of this technology point, maybe if we get it right, maybe it might be even better. It's a good point. Second thing, and I don't completely agree. Now, so far as we have studied that, you're right, not many people have a runny nose and so on, but as you might have noticed, and everybody in the room have noticed, one side is always clogged and the other one is usually open, unless you don't have a completely a rhinovirus infection or things like that. So that's, I think it's a good point, because we were actually thinking of might not be one nostril enough, but after starting that one nostril, even if one nostril is blocked, the other one is free, we decided to go for two nostrils.
Thanks. We have time for one more question? Yep. Is there one more question? There's one back in the back.
One more here.
Jim Baker, Dan, Ohio. We did, I'm sure many of you did performance art in BARDA's conference room last summer and fall. And judging from the response that they had to every partially approved vaccine, if we could call it that, and the documented need for vaccine that wasn't there in the fall, do any of you feel that there will be any situation where we will have approval for a vaccine that hasn't gone through, as you alluded to, the red zone, and full approval by the FDA?
Lots of people been there, done there, and they got the T-shirt. You know, there's really no shortcuts at FDA. I've been doing this for over 40 years, and we're not going to get that. You have the emergency use authorization, and that's, in theory, a pretty simple way to get a vaccine approved. But, you know, where are we on that? It's a very risky thing. You have to do a lot of post-marketing studies, and it's a very difficult thing to prove. You have the accelerated approval mechanism right now in place for flu vaccines, and that's about as good as you're going to get. You know, the FDA hasn't got a lot of regulatory agencies around the world don't have a lot of success with slipping things through, you know, greasing the path. And remember, it's a risk-averse agency, and they're supposed to be there, number one, to protect us, and second, to get drugs approved that help us. But number one is do no harm. Remember that. So I don't see that happening. I do think that in terms of, just to follow on sexy delivery, there's so many sexy ways to deliver a recombinant protein. Holy cow, people come in our door every single day. There are so many ways. The patches, the ocular, there's a nice H5 receptor in your eye. You put a little drop in your eye, a little spray in your eye. Intranasal, oral, there's almost so many ways to do it. But one thing I learned over many years is that rule one of new pharmaceutical product development, never have two unknowns in a new product. So no adjuvants for us, no fancy delivery stuff. We're going to get it approved through an injection. After that, man, it's Katie bar the door, because there's fantastic ways to get your vaccines delivered that will revolutionize this field.
Dr. Adams, we'll give you the last word on that. Yeah, well, maybe not. Would you, Dr. Singhvi, did you?
Well, I just wanted to echo what Dan said, because I think the one part that I think we are missing in this whole new technology stuff is that this is the regulatory path, and it is not the sexy science that matters. It is whether you can make the product consistently. It's having enough safety data. It's having the ability to show that you can make multiple different types of strains, and doing a lot of validation and a lot of characterization of your product, a lot of analytical work. So when the rubber hits the road, it's really hard to get a new vaccine approved. Very, very hard. So this is a tough business, and the red zone, or whatever you want to call it, when you get into that zone, the hype is over. You got to you really have to do some really hard work, and you have to put in a lot of money in this.
Thank you. I'd like to thank all of our panelists for, I think, an extraordinarily stimulating discussion.